Thank you for the opportunity to be with you today. So the optical microscope was developed first in the 17th century, and after 50 years of improvement, microscopists like Hooke and van Leeuwenhoek were opening up an entire new microworld of living systems beyond the ability of what the naked eye could see. Microscopes steadily improved until the end of the 19th century through oftentimes a lot of people doing experiments and building microscopes themselves, oftentimes with fits and starts and a lack of reproducibility. But then a microscope manufacturer, Carl Zeiss, in the 19th century, teamed up with Ernst Abbe, a physicist, and Otto Schott, a chemist, and really made a science of microscopy and figured out exactly how images are formed in the microscope and how to create lenses to make that happen. One of the consequences of this was that Ernst Abbe discovered that there was a fundamental limit to how small you could see in the optical microscope that was half the wavelength of light. So because they were able to make a reproducible scientific instrument, optical microscopy became ubiquitous in the 20th century. In fact, it is so ubiquitous today that if you did the deletion test and erased optical microscopes, there's no branch of science and technology that would not be impacted by the lack of optical microscopy. But at the same time, because they perfected the microscope at the end of the 19th century, in some ways it stagnated. And so the microscope you could buy from Zeiss in 1880 was really not all that different from the microscope you could buy from them in 1980. There were additional contrast mechanisms and so forth, but it was basically the same instrument. So as a result, in the 20th century, it was really the developing fields first of biochemistry and molecular biology that really became predominant. And it's been so successful that today most biologists have a worldview where they look at living systems through the eyes of biochemistry or molecular biology. But that started to change starting around 1980 thanks to unrelated technical innovations, a lot of them from quantum mechanics again. First was the transistor, which led to the personal computer so that you could control microscopes automatically. Also, you were able to create sensors that could detect light very efficiently. The next was immunofluorescence technologies, whereas instead of seeing ghost-like images in black and white of cells, now you could illuminate any one of the tens of thousands of different types and proteins specifically and see them individually. That had another big boost in the 90s with the development of fluorescent proteins, so you could coax the cell to do this by itself and do it in live cells. And the third was the development of lasers, which allowed us to illuminate these fluorophores very precisely and illuminate a number of them in the same cell at the same time. So as a result, if you stir those technologies together in different ways, there's been a Cambrian explosion of microscopy technologies since the 80s. First with the confocal microscope, then spinning disk confocal microscopes that look at live cells, 2-photon microscopes to look in scattering tissue, super resolution microscopes like the ones I developed to see beyond Abbe's limit, light sheet microscopes that can look at 3D dynamics, and adaptive optic microscopes that can look into aberrating tissues. So what these microscopes have revealed is the cell is incredibly complex and incredibly dynamic. And if you don't take into consideration the fact that it's the dynamism that really defines life, you are missing a very important component of what's going on. And so I'll give you a couple examples of that. So here, using a super resolution technique that I had a hand in developing, one of our collaborators looked at the Huntington protein, which is involved in Huntington's disease. And what they were able to show is here you see two live cells, the nuclei of the live cells. At the left, the Huntington protein is wild type, and so it just creates very small and transient aggregates. But when you have the glutamine repeat of the mutation in the disease, you get these large aggregates in the nucleus. Now, these aggregates are endemic in lots of different diseases, and they've been known for a long time. But what our collaborators were able to show was that when they co-labeled various transcription factors, at the bottom right, you're seeing individual transcription factor molecules, and when they meet these aggregates, they get bound up in them like they're caught in quicksand. And so it suggests a potential possibility, an unthought of possibility, that maybe you're getting down regulation of gene expression later on because its transcription factors can never get to the DNA to produce the RNA. Another example is, this is a textbook picture of a cell. Normally you go to a class and you hear, oh, well, the ER is where we make the proteins and the mitos are where we make the energy of the cell, and you think of them as individual entities. But the reality is not like that at all. So this is another high-speed super-resolution microscope looking at the endoplasmic reticulum and the cytoskeleton in microtubules, and the microtubules are constantly remodeling the endoplasmic reticulum, and also these degradation lysosomes are pulled along the microtubules to also push the ER around and remodel it, which also has influences in other organelles. So if we do the same type of experiment in 3 dimensions, we see that with 6 different organelles labeled, that the contacts between organelles are ubiquitous, particularly the contacts between mitochondria and the endoplasmic reticulum, and those act as organizing centers for other organelles to come and dock temporarily for biochemical reactions. So the cell isn't a bunch of individual components, it's a bunch of constantly interacting components with transient kiss and run interactions happening everywhere, which are essential to its function. So the problem with the techniques I showed thus far, though, is that we're looking at cells in isolation. And biologists know that the phenotypes that we observe are the result of gene expression. And gene expression is partially influenced by the local environment. So if you look at an immortalized cell on a cover slip, you're far from where the cell evolved. And so you need to look at these systems truly in vivo and where they evolved. So we borrowed the techniques of adaptive optics from astronomers to be able to clear how our light goes in and out of the specimen, to then look at this dynamism in full in vivo systems. And as you can see in the top right case, where you're watching the dynamics of an immune cell as it's going through the inner ear of a fish, that type of story, how all those cells are moving, each of them, every one of you, have 37 trillion cells where that type of choreography is going on constantly. Each one of those cells has several billion protein molecules which are interacting in the same way as in that Huntington's example I showed earlier. There's more complexity in there than there are stars in the universe inside of one body. Inside of all the brains of humanity, there are more synapses than there are stars in the universe. So we have the most complex matter that is known in the universe is right here in front of us. So all of that's the good news in terms of the development of these tools. The bad news is trying to make all of these tools accessible. And this goes all the way down to the most simple microscopes, the microscopes that could have an impact in third world countries, and particularly for the diagnosis and treatment of infectious diseases. So all of these diseases I'm showing here, although you can diagnose them by their phenotypes in the individuals, if you want to understand what strains are involved and how severe it is, you would like microscopes to be able to do that. That's what we do in the more developed countries. And a number of groups, particularly over the last decade, have taken on the challenge of making extremely inexpensive microscopes to be able to use in the third world. The fold scope, for example, from Manu Prakash at Stanford is less than $1. It's very much like Van Leeuwenhoek's old microscope hundreds of years ago. But there's many, many problems such that, as far as I know, none of these have had any kind of significant impact inside of the third world despite considerable effort. And it isn't just the technology. There's so much additional infrastructure and human things and so forth that have to get involved to really make an impact. And I think all of these problems are solvable, but I think that they require a concerted effort by a number of different communities. So there's an analogy on the opposite end of the technological spectrum with the microscopes we make. The problem with these microscopes is, although they're very potent in what they can reveal, they're very complex and very expensive. And the problem is that, you know, we want these to be in the hands of biologists, but there's many biologists in the world. There's very few microscopists who can build these microscopes in the world, and the Venn diagram of the fraction of those that can get together and actually build and then use these microscopes is even smaller. So like with one of our more successful microscopes, we've worked as hard as we can to try to make it so that we can disseminate that technology to others. We've probably in 4 years gotten 90 groups to start to build these microscopes, and maybe 20 are now operational. So it's a very difficult challenge in order to get these things going. Ultimately, the only way biologists are really going to use technologies like this is if they can be commercialized and made turnkey. You know, you heard from Helen's talk. They have many, many other things to worry about than whether their microscope is working properly. And so they need something that works. And to do that, you need basically market forces to put in the energy and the product development necessary to make it commercial. And this is a common thing where you have this valley of death, of where you go from a research idea to commercialization. And there's a chicken and egg problem because the companies aren't going to want to invest the money to make these types of microscopes until they know there's a market. And the biologists don't know there's a market, that it's useful until there's a useful microscope for them to work with, so they don't tell them there's a market. So to get around with that problem, at my institution, we developed an advanced imaging center where we have these pre-commercial microscopes, and then for free, anyone around the world can apply and come and do their biology on our microscopes, and we help them with data analysis and so forth, and then they go on their merry way. This has been very, very successful, and it's been successful enough to get a couple of these microscopes actually in the pipeline at companies for commercialization. But there's additional problems beyond that, and the first is that just as you can't build a car that can take the kids to daycare on Friday and then win at the Indianapolis Speedway on Sunday, you can't build one microscope that can do everything. Microscopes have to differentiate. That's a problem because if you have an imaging core for biologists, that means they need to have 6 microscopes there, and they don't know which one the biologist is going to need on any given day, so a lot of them are dark. So the last project I'm doing at Janelia is to basically make a Swiss Army knife microscope that takes almost all of those technologies I told you about earlier, this Cambrian explosion, and put them in one 4-foot by 4-foot box at a reasonable price that, again, initially will only be those people in that little bit of the Venn diagram that can build it, but hopefully these ideas will eventually hit the commercial sphere. And then imaging cores can have a suite of identical microscopes, push a button, and this biologist can figure out which works for him, and this one can figure out which works for him. The bigger problem that we face now with our most modern microscopes is that they produce terabytes of data per hour. So we have a paper that was just accepted that has 1 petabyte of data that was analyzed for it. And so most people do not have the computational resources to extract the meaning. You can create beautiful movies of animate cells, but you're not going to get all of the understanding of how these transient interactions are happening in the cell without massive computational resources. And so I've recently moved to Berkeley, and we're trying to attack that problem with the Next Generation Imaging Center with a specific focus on trying to deal with, in an integrated way, between the biologists, the data scientists, and the instrumentation developer, how to attack this problem of developing open source algorithms so that people can get quantitative biological insights from these massive data sets. And so to conclude, I'd just like to say that again, in the 20th century, the way, I come to biology from an outsider since I'm a physicist. And the way I look at it is kind of like the old paradigm of the elephant and the blind men. And so you're trying to understand living things, and the molecular biologists are holding the tail, and the biochemists are holding the trunk and perceiving one thing or the other. But although electron microscopy has played a role in structural imaging, the thing that defines life is that it's animate. And so the only way we're really going to understand life is to understand these dynamic interactions. And thanks to this explosion that's happened since the 80s, we now have the tools to make this actually a trinity in terms of what we would be able to understand. And maybe we don't get the whole elephant yet, but at least we can hold another part of the elephant in order to be able to understand biology in a new way. Thank you.